We have been discussing the electrophoresis. It's used repeatedly in the molecular biology course. Now, electrophoresis, as a matter of fact, is the working horse for the molecular biology. Today, in this segment, we will discuss or we will study what are the different support media for electrophoresis technique and how we can use that support media. Now, the first is why there is a need of support media. The thing is that the adverse effects of diffusion and convection current that was required to be minimized by stabilizing the medium. If you recall, in the earlier lecture, whenever we do the electrophoresis, under the charge, there are present some convection currents which are due to the electric current, and also there is a temperature rise of the media. So it means that these two things, they adversely affect the result of electrophoresis. And in order to counterbalance these effects, we need some sort of the support media on which to support the molecule. Now, stabilization, that was achieved by carrying out electrophoresis on a porous mechanical support that is wetted in electrophoresis buffer, in which electrophoresis of buffer ions and sample that could occur. So the one idea was that we can stabilize the compound or stabilize the electrophoresis by carrying out electrophoresis on a porous medium rather than on a gel, or rather there should be a medium which should be porous but which should be at the same time supportive to the molecule. And the support medium that cuts down the convection current, that is where the convection current which are there, that it should also be capable of cutting that down so that the separated compounds, they remain in the sharp zone. That is, they shouldn't diffuse with each other. So that should be also the characteristic of the support medium. The earlier supports medium which were used, they were filter paper or cellulose acetate strips. They were wetted in the electrophoresis buffer. That is, the filter paper as used, their sample was put on that and wetted in the electrophoresis buffer so that they can support those compounds or those separated compounds separately. Or we can also use the cellulose acetate strips which were used conventionally earlier. Now, small molecules like amino acids, peptides, and carbohydrates, they were routinely separated and analyzed by electrophoresis using these supports conventionally for a long time. Now, such molecules, they are now more likely to be analyzed by more modern and sensitive techniques such as high performance liquid chromatography. Now, HPLC, as the name stands, that is a more precise technique which can be used to separate these compounds. Now, paper on the thin layer supports that is fine for resolving small molecules. When we use paper as a support, then they are fine for resolving small molecules, small molecular weight, and the separation of the macromolecules is also can be made by this technique. Now, another choice as a support medium is the use of gels. You see the gels, they will have different density, different porosity. So the introduction of the use of gel as a support medium, that led to a rapid improvement in method for analyzing the macromolecule. You see, we can prepare gels of different molecular weight, of different porosity, and they will keep the compound or separated compound, the separated zone, very clearly differentiated from one another. Now, in that case, we can also use a starch gel, that the gels which are earliest gel system to be used, that was the starch. Uses starch is also makes gel, and the compound can be put into it and electrophorise. But the use of gels, the majority of electrophoretic technique, they use nowadays either agarose gel or the polyacrylamide gels, which are more sophisticated, more modern ways to use. Advantages of this is that by using different concentration of agarose gel or the polyacrylamide gels, we can prepare the gels of known porosity, of known support characteristic, and they will definitely give the separation beautifully and very clearly in the clear zone for studying and for separating the molecules, macro or small molecule with which we are working with. So these are, as a matter of fact, the support medium for the electrophoresis and how they are evolved and how they are used for this electrophoresis technique.